Good day everyone, it's me Joey Sal and let's learn about photosynthesis. We talk about cellular respiration transform the food we eat into adenosine triphosphate. But what do plants do? They don't really eat in the normal sense of the word, apart from Venus flytraps. As it turns out, plants are getting their energy directly from the source, the sun. Plants use sunlight as well as water and carbon dioxide, which are plentiful in our earthly surrounding, to produce their own nourishment through cellular process called photosynthesis. This word makes sense because photo comes from the Greek word light and synthesis means to build. So plants use the light and small molecules to build bigger molecules that they can then metabolize to produce energy. Other organisms then eat the plant and other organisms eat those organisms. So plants really are the foundation of the entire food chain. Let's learn about how photosynthesis works. Photosynthesis consists of two stages. There are the so-called light reaction in which the solar energy is converted into chemical energy, and then there's the Calvin cycle which uses the chemical energy to build G3P, the precursor to glucose and other molecules. Both of these stages occurs inside one of the organelles found in plant cells, which are called chloroplasts, and there are around 30 to 40 of them per cell. A chloroplast has two membranes surrounds a fluid called stroma, within which sacs called thylakoids are suspended. These sacs are often stuck in column called grana, and the membrane of these thylakoids sets a special pigment molecules called chlorophyll. Chlorophyll can take either an A or B form, differing on in one function group inside the porphyrin ring, either a methyl or the aldehyde group. The porphyrin ring is what gives these molecules the capacity to absorb sunlight in a unique way. If chlorophyll absorbs a photon of a particular energy, one of its electron jumps up excited state. These chlorophyll molecules set inside of a photosystem with many other chlorophyll bands. This excitation can get passed around from molecules to molecules with one electron relaxing back to the ground stating emitting another photon that will excite an electron in another molecule, and so forth like pinball machine. Eventually, a photon may strike the reaction center complex, which also contains two molecules of chlorophyll. When this happens, the high-energy excited electron, rather than relaxing back down to the ground state, will instead be transferred to another molecule called the primary electron acceptor, after which an enzyme supplies the missing electron to oxidize chlorophyll via a water molecule to get it back to normal, which is how oxygen molecules are produced. This is the first step of the light reactions. These are the series of redox reaction that occurs after photosystem 2 and this in photosystem 1. They are named backward. It's because they were named in order that they were discovered. In photosystem 2, electrons flow beginning with the primary electrons acceptor through a series of compounds that are embedded in the thylakoid's membrane, like the electron transport chain from the cellular respiration. Adenosine triphosphate will be a byproduct of this process due to a proton gradient and ATP synthase. So just like in cellular respiration. Now electron moves through photosystem 1. Now electrons will move through photosystem 1, another electron transport chain, but instead of producing ATP, photosystem 1 will result in conversion of nicotinamide dinucleotide phosphate or NADP plus into NADPH. So, the light reaction in the thylakoids, which are summarized by the following equation, require sunlight and water among the reactants and generate oxygen, ATP and NADPH as products. ATP and NADPH are then used in Calvin cycle, which occurs in the stroma. This is where all synthesis occurs. Unlike the citric acid cycle, which is catabolic, the Calvin cycle is anabolic, building all organic molecules from smaller components requiring energy to do so. This happens in three phases. Phase 1 is carbon fixation, which is catalyzed by an enzyme called ribosco. Ribosco captures CO2 or carbon dioxide from the atmosphere and attaches into a 5-carbon sugar called ribulose biphosphate. The resulting 6 carbon molecules is unstable and splits into 2 molecules of 3-phosphoglycerate. Phase 2 is reduction. Each of these molecules receive a phosphate from ATP and is then reduced by NADPH, after which it loses phosphate to become glyceraldehyde phosphate. Some of these G3P is the output of Calvin cycle, going on to other pathways that generate glucose and other organic compounds, and some goes back to generate some RUBP and start the cycle over again. To generate a net output of 1 G3P, the cycle needs 9 ATPs and 6 NADPH.